Hi, I'm Joel Glover from the Norwegian Center for Stem Cell Research. An important issue in stem cell research is the design of in vivo systems for testing the differentiation potential of stem cells. In this video, my colleagues and I will demonstrate how to use the chicken embryo as an in vivo host for xenotransplantation studies of human stem cells. Fertilized eggs are allowed to develop in a warm, humidified incubator until the embryos have reached the desired stage. First, wash the eggs with 70% ethanol. Then remove about 4 milliliters of albumin with a syringe and an 18-gauge needle to create an air pocket over the embryo. Avoid piercing the yolk during this process. The air pocket is necessary to avoid harming the embryo when opening the window in the eggshell. Seal the hole made by the needle with conventional plastic tape. The next step is to cut a window through the eggshell over the air pocket. To strengthen the eggshell and to prevent eggshell particles from falling onto the embryo, place tape over the air pocket, and then cut through this using a sturdy dissection scissors, staying within the bounds of the air pocket. Any air bubbles present over the embryo can be popped with a blunt probe like the back end of a pipette tip. To contrast the embryo, prepare a 0.1% fast green solution in chicken